Okay, I'm going to change animal or plant kingdom to animal kingdom here. The point I'd like to start with is that animals have evolved to any ecological niche where we would want to operate a robot, except for maybe outer space. So they are proven models for investigation, obstacle negotiation, search, the sorts of things that people really want robots to do. What we've been trying to do is to find ways to engineer devices that capture the performance capabilities of animal models. We use invertebrate model systems predominantly, although we do some work on sea lamprey, which are a lower vertebrate. The beauty of invertebrates is that they have a distributed nervous system. They have a segmental body plan, and there's a series of mini ganglia that control each of the body segments. One ganglion in particular, which is called the stomatogastric ganglion, which controls the movements of the stomach, is shown in this slide. And each of these round objects here is the cell body of a neuron. And these neurons can be rigorously identified from animal to animal. They have names. And we can penetrate them with microelectrodes and record their transmembrane voltage. And when you can record from pairs of identified neurons, you can look at their direct electrical interactions, which are called synaptic connections. And if you look at all possible pairs of two neurons in this ganglion, this is the circuit that you see. This part of the circuit generates a slow rhythm that has a period of about 10 seconds, and this part of the circuit generates a fast rhythm that has a period of about a second. And these cells down here are modulatory inputs to this ganglion that modulate it to produce a broad variety of behaviors. Now, we are able to do multiple intracellular recordings from these neurons, do ionic substitutions, do perturbation experiments where we stimulate with inputs at different phases in the rhythm, and finally do experiments with what are called neuromodulators. And right now there are about 130 known compounds which not only alter the properties of individual neurons, they alter the connections between the neurons. And if you've heard people discussing over the last few years something called the connectome, there really is a family of connectomes. And this ganglion, which has only 30 neurons, has this level of complexity. And when people start telling you that they understand how the cortex is working, it's borders on what I call fancy. Now, after about 40 years of study, we've established a general architecture called the command neuron, coordinating neuron, central pattern generator model. And this applies to all organisms that exhibit bilateral symmetry and explains pretty much all what we call innate behavior. Now, when I say innate behavior, I'm talking things like walking, chewing, breathing. We can learn to speak in Italian or German or Chinese, but we all walk in human. Okay, so these are innate behaviors. And most simple animals show only innate behavior. They don't show any form of learned behavior except rapid food aversion. Now, these central pattern generators in multi-limbed animals are coordinated among themselves by a category of neurons called coordinating neurons. And then they're activated in turn by a category of neurons called command neurons, which descend from the brain and turn these networks on. Now, command neurons are the locus at which the decision to evoke a behavior is made. And in animals like lobsters and crayfish, you can dissect the brain connectives and pick out single axons. And some will cause forwards walking, some will cause backwards walking, some will cause feeding, some will cause swimming. They represent the least divisible unit of behavior, the behavior that's evoked by a single command neuron. Now, I say this as a general model, and you can take these same components and rearrange them and form a central pattern generator for a swimming organism such as the lamprey, which is another animal model we use. Now, what we do in my lab we call biomimesis, and we try to go from animal models to robots. And this is our second lobster robot. And this is really quite unique because it has a biomorphic plant. The claws and tail are hydrodynamic control surfaces. It has a neural circuit-based controller. It has neuromorphic sensors that code in a labeled line code, artificial muscle, and it has a library of behaviors that were reverse engineered from real lobsters. The other animal model that we use is the sea lamprey, which is an undulatory organism. And this robot is made out of entirely the same components as the lobster robot. In fact, the software modules are just slightly modified between them. But what came out of building these two robots was the realization that if you try to control a robot algorithmically with computer programs, unless you've anticipated every possible situation the robot gets into, you're going to get into situations where you don't have an escape strategy and it's going to get stuck. And I can guarantee you that about 99% of robots that exist right now have this deficit. Now, animals never get stuck. What animals do is they wiggle and squirm when they get into tight spots. And we think that what the animals are doing is that they're increasing the level of chaos in the networks that would otherwise generate locomotory movements. And the chaotic variations on the locomotory movements are the wiggling and squirming. So the idea is, can we build robots that can show wiggling and squirming? And our solution to this is to try to use electronic nervous systems. But the problem with electronic nervous systems is that they have to be able to operate in real time. Now, I went on a sabbatical to the Institute for Nonlinear Science, and this is a very interesting place that consists of neurophysiologists and physicists. And the physicists asked the neurophysiologist, how many degrees of freedom does a lobster neuron have? So one of my colleagues, Alan Selverston, took this ganglion and isolated one of the cells from all sources of synaptic input. And sure enough, though every neuron he looked at, and this is the same neuron in 4 different preparations, showed truly chaotic activity. And Henry, who is an expert on nonlinear dynamics, got Al to do some experiments where they fooled around with the inosine triphosphate calcium buffering system. And when the inosine triphosphate system was suppressed, they got rhythmic bursting. And when they increased the inosine triphosphate system, they got chaotic activity. So Henry developed a form of analysis called local false nearest neighbor analysis and determined that in the rhythmic pattern, the system had only 3 degrees of dynamical freedom, and in the chaotic pattern had 4 degrees of dynamical freedom. So this greatly simplifies the dimensionality of the models. So another colleague named Sasha Volkovsky worked with some equations called the Hindmarsh-Rose equations, which describe the interrelations of these 4 dynamical degrees of freedom. And they were able to realize this as a 4-integrator analog computer. Now, the beauty of analog computers is that they operate in real time. So if you use analog computation, you can build robots that operate with the speed of, you can build computers that operate with the speed that a robot needs. And then they instantiated these as circuit boards. And this is an electronic neuron. And when you plug this into an oscilloscope and record X, which is a transmembrane voltage, it's pretty much indistinguishable from a living neuron. And I've done all sorts of perturbation experiments, and these really, really have the true dynamic behavior of real neurons. What's interesting is that there's one parameter here which we can control with a trim pot called E, and if you increase E, you can switch this neuron from rhythmic bursting to chaotic. So these are capable of variable chaos, and we can vary the chaos by just varying one internal voltage. So I took a circuit that I worked out years ago for the control of walking in different directions. And the basis of this circuit is that there's a central pattern generator that activates motor neurons, and there are some command neurons for forwards and backwards walking that gate some particular synapses within this circuit. So we wired a bunch of these boards together and created this circuit, and sure enough, we could produce the motor patterns that you would see in a behaving lobster for walking in forwards here and backwards walking. Now, these signals are not particularly good for controlling motors, but they're ideal for controlling muscle. So we found a material called a shape memory alloy. And the shape memory alloy has 2 stable crystalline states. This is the length of a wire versus temperature. And if you heat it to a very high temperature, it goes into a dense crystalline state called austenite. If you cool it, then you can mechanically deform it into another state called martensite, and this is associated with about a 5% length increase. If you then heat it again, it will contract into martensite, and a 250 micron wire will lift a kilogram in about 150 milliseconds. It's one of the most force-dense materials known. It's what they use to make heart stents out of. Now, what we do is we take pairs of wires and we activate them with trains of motor neuron activity from these neurons. And the way we mediate excitation contraction coupling is we take the action potentials from the electronic neurons, we gate them through a comparator, and then apply that current pulse to a power transistor, which applies current to the nitinol to heat the nitinol. So when we're heating one wire and making it contract, we're stretching out the opposing wire. If we then take one of the legs of my robot and wire it up to a network of 8 of these neurons, we can get it to walk in different directions. And this is done entirely with analog circuits. There's no algorithms, there's no digital computer here at all. So this is really a revolutionary way of controlling robots that's never existed before. Now, the problem with these boards is they're big, and if you need hundreds of them, you're not going to be able to stuff them into the hull of a robot. So one of my other colleagues, Nikolai Rulkov, came up with a phenomenological model based on a map. And this is the membrane voltage in cycle n plotted versus the membrane voltage in cycle n+1. And he has 2 equations for x and y that describe the fast component x and the slow component y. And there's some fuzzy logic over different ranges of x. And there are 2 control parameters called alpha and sigma. And in the space of sigma and alpha, in this area, the neurons are silent. In this area here, they show action potentials at constant frequency. In this area, they show bursts of spikes. But more importantly, in this yellow area, they show chaotic activity. The beauty of these models is that they're based on difference equations, and we can get about 2,000 neurons and synapses to operate on a single DSP chip. So we can achieve this goal of controlling a robot with a nervous system with variable chaos using this technology. We have similar math for the synaptic currents, so we can model synapses, and we instantiate these in LabVIEW. And LabVIEW is the programming environment of LEGO Mindstorms. So we have a module that you can attach to LEGO Mindstorms that will let high school kids control robots with electronic nervous systems, which looks good on the broader aspects part of an NSF proposal, I can guarantee you. So this is my 2nd generation lamprey robot, and this is controlled with about 400 neurons and synapses. And this is the central pattern generator from one body segment. And these excitatory interneurons would activate the motor neurons. Now, if we wire these together in a series, so there's a central pattern generator for each body segment, and they have these ascending and descending connections, if we bias the excitation towards the anterior segments, we get front to rear propagation and forward swimming. If we bias it towards the rear, we get rear to front propagation and backward swimming. This is RoboLamprey in operation right now. This is the current version, and this will swim at about 40% of the maximum theoretical velocity of a lamprey. So we're getting really close to having the same performance as the real model. Now, what's really important is to be able to get this thing to show adaptive behavior in natural environments. So we have a circuit board that has a sonar digital signal processor on it, a compass, and an accelerometer. And these are some of the primary senses we use in guiding the navigation of this system. What the sonar system does is it has 3 microphones on the front and a signal processing system. And what it computes is the deviation of a target here from the main axis of the hull, both in azimuth and inclination. And it codes this in terms of interneurons that fire in proportion to the deviation of the target from the hull heading. And then for impediment, with these things are swimming along, with the accelerometer, we can detect collisions. And the collisions are a high amplitude deceleration. And we can get that to code as an interneuron that will release an avoidance reflex. And then we've developed a neuronal compass, which takes the actual heading and compares it with a desired heading, and then codes this in the discharge frequency of what we call heading deviation neurons. And as we rotate the robot through 360 degrees here, this is the actual heading, you can see that it biases the output. So here it's at a big deviation. There's a big asymmetric bias on the output to the 2 sides, and as we get close to the normal heading, that levels out. So we have a way of giving this thing a sense of direction, which is quite important. Now, the brain of this thing consists of a series of command neurons that activate on each of the central pattern generators. And on each side, there's one for backward swimming, for slow forward, for fast forward, and amplitude modulation, which is used in steering. And then we organize the inputs from sensors into what we call exteroceptive reflexes that in turn act upon these command neurons. So these are the sensors, and these are the interneurons that form part of the brain of this robot. Now, I want to show you an experiment where we interact two of these reflexes. So what we're going to do is change the desired heading of the robot, and it's going to try to turn into that desired heading by activating these heading deviation neurons. And then while it's changing direction, it's going to collide with a rock, and it's going to superimpose an avoidance reflex on this heading change. So this is this reflex in operation. So we're telling it to swim in this direction. So we've just turned it on, and as it's turning in that direction, it's going to run into a rock, and then it's going to back up. And as it's backing up, it's going to be altering its heading to get on the desired heading. And this is all done with interneurons. So there's no algorithms in here at all. So we say we program in neurons and synapses and not computer algorithms. So now it's going to head off on its desired heading. And it's done all that with network control without algorithmic control. Okay, now this is our other robot that we're working on. This is a new version of the lobster robot, and this is kind of just about to come together. We've got it operating with these now analog neurons to walk in different directions. We have a board set that we shared commonly between the lobster and the lamprey robot. It all uses the same hardware, and we're in the last page parts of getting all this stuff to fit together in the lobster robot. And it has a similar set of reflexes for heading deviation, for collision, for optical flow, and for input from its antennae. And this is basically the brain for the lobster robot to give it pretty much the same capability that we see in the lamprey robot. Now, the beauty of these things is that they're very simple electronics. And in fact, the most expensive part here is the processor, which is $29. And then we have some very cheap parts. These two parts for each actuator cost 81 cents. And then the nitinol actuator costs about 12 cents. Now, in comparison, a PID controller for a motor would cost about $400, and the motor would cost about $200. So, and given that we have 48 degrees of freedom, you can see how expensive this could get very, very quickly. So this is a very, very cheap solution. We have similar input for the sensors, and we've created a new term for the Navy vernacular, which is cheap robots. And really the most expensive part of these robots when you get down to it is the batteries, which is pretty remarkable, outside of the intellectual capital. Now, people always ask me how we would use these things, and what these are not good for is going long distances. So we would put these things in a mothership we call the Stealth Zodiac, and this would have in it a gantry crane and a well that we could deploy and retrieve the robots from. And then this robot would have a sonar beacon that we could use to retrieve the robots, and then we would put a heterogeneous swarm of crawlers, swimmers, and then a third robot that we base on the abalone we call RoboLobster. And this is a static sensing and communication node. And in use, these would all communicate with sonar underwater, and the RoboLobster would form a long baseline navigational array so we would know where these things are. And the RoboLobster could deploy a GPS antenna so you would know in absolute coordinates where each of the robots is. And then the basic algorithm for search is what we call a search vector. And that consists of a compass heading, some odometry information, and a propensity to either negotiate obstacles or investigate them. So we would send this thing on a search vector, it would enunciate its position, it would be given a new vector, it would go off and search randomly. And this is really important on contoured bottoms, because if you try to do lawnmower search patterns, which is what almost all robots do nowadays, the minute you hit an obstacle, you're screwed up. You got to reset. Okay, we want to get these things to show group behavior, and group behavior is really well known among arthropods. This is queuing in lobsters. But one of the more interesting ones is a phenomenon called stigmergy. And stigmergy is best illustrated in nest and hive construction in social insects. Now this is the nest of a fire ant, and this is a pretty young nest, and this is an older nest, but you know notice they have a very stereotyped architecture, where there's a helical central gallery, and then there are these side chambers that have pretty much the same dimensionality and spacing. And the thing that's interesting about this is that there's no architect and there's no plan. This results solely from low-level interactions between the individuals, and these interactions have to take the form of exteroceptive reflexes, which is the basis of the architecture of all of our robots. Now, the 800-pound gorilla in stigmergy is that much of the local low-level interactions between the animals are based on chemical cues. So ants, for example, have 19 pairs of glands that produce different scents that they use in different situations. And crustaceans actually communicate by urinating on each other. And if you inject fluorescent dye into their bloodstream, you can show they're interacting with fluorescent dyes. So trying to get chemical sensing into these robots is really, really important. Now, just dealing with social insects, we have another project which is to build robotic bees for pollinating crops. And I want to show you how this same architecture, which was originally developed for underwater robots, also works for flying robots. And we get 45 milligrams on this vehicle to build the brain, which is kind of a challenge in and of itself. So we have to find a really simple way of controlling this beast. Now, bees operate on search vectors, and the search vector is communicated stigmergically by what's called the waggle dance. And the bees do this waggle in a queue, and this indicates the orientation of a food source relative to the hive via a sun compass, and the intensity of this waggling conveys the odometry information. So to get a search vector and a bee, we need to be able to communicate those two forms of information. So we have these heading interneurons. I've described those already. And we do this by a simple computation of between the desired and the actual heading. I didn't realize I had so much action in this slide. But the hard part was the odometry. And it turns out that bees use optical flow, the actual motion of the visual surrounds past them. And one of our colleagues, Jeff Barrows, has developed a single chip optical flow sensor. And so what we do is we take the optical flow, and that's coded as a spike train, and then we have a synapse onto a second order neuron, and then a third order interneuron. And it turns out that by varying the strength of the synapse, we can get this third neuron to fire at different odometric distances. And so we use the modulation of synaptic strength to code the odometry information. And the modulation of synaptic strength in nervous systems is mediated by neuromodulators. So it's a very standard form of integration. Now, controlling the motors, initially we're doing this with helicopters, and the helicopters we use have two rotors, a clockwise and a counterclockwise one, and they're controlled by pulse width duty cycle modulation. So we can take the output of a flight interneuron and use it to code this pulse width duty cycle, and so the variations in the discharge frequency are going to code for different rotor speeds. So if we have two interneurons for clockwise and counterclockwise rotation, and these are activated by optical flow neurons. If the animal is flying, translating in a straight line and gets blown off course, this eye is going to see translation plus optical angular rotation. This is going to see translation minus angular rotation. This is going to bias this more than this one and bring it back on course. So that's the minimal circuitry you need to keep it flying on a straight line. But we can superimpose our heading deviation neurons over this to override that in case it really gets off course. And then we can use the optical flow information to activate the odometer. So with this minimal circuit, we're already getting the helicopter now to be able to do search vectors on a compass heading and get the right odometry or go for the proper distance. Now, what you're probably all interested in is what this has to do with synthetic biology. And at this point, I thought I was doing synthetic biology. We called it synthetic neurobiology. And there was this advertisement for a thing called a sandpit. And several of you were attendees of this event in which we were asked to take on some grand challenges of synthetic biology. And so we were asked to form teams, and I formed a team with Chris Voigt, Vlad Parper, and Dan Frankel. And the two challenges we took on were, can we get engineered cells to talk to microelectronics? And two, can we find a way to couple power with actuation? Because these are really two of the bigger problems in robotics. And if we're going to address this stigmergy problem, we've got to figure out a way to get olfaction into robots. So the idea was to build an undulatory vehicle loosely modeled on the hatchet fish. And the idea was that we would have engineered sensors that would talk to an electronic nervous system, and the electronic nervous system would activate engineered muscle. And originally we were planning on powering this with microbial fuel cells. And it turns out the microbial fuel cells can produce enough power, but what they don't do is regulate the power well. And the circuitry, the subthreshold analog VLSI, depends on highly regulated power, and that just wasn't going to work. And we couldn't get enough power to activate the muscle. I'll show you how we do that. So we're going with a thin film battery at this point. So at least my take on synthetic biology is you start off with parts and then you build devices. And the kind of device we wanted to build on the sensor end was to get a G protein coupled receptor. And the kind of holy grail in the robotics world is to get one that can respond to things like RDX. Couple that with a calcium channel such that when this smelled something, calcium would enter the cell, and then couple the calcium channel with aquiorin, which is a photophore that gives off light, so that you could basically see smell. Now, the alternative into this is the output end, in which we take fibroblasts, and we're putting in the genes for channel rhodopsin, a controller gene, and then what's called tree muscle red construct, which converts the myoblasts to myofibrils. And this is work that Vlad Parper has done. And the basic idea is that we insert these constructs into fibroblasts. We use antibiotics to select for cells that have the construct. We use a DOX repressor to hold these in this state of myocytes. We can then form these into whatever we need on the robot, and then remove the DOX and transform the myocytes into myofibrils so that we have active muscle. The beauty of this is you can hold this for a long period of time in the fibroblast state, so you don't have to worry about how you keep muscle alive for long periods of time. Now, the hardest part in artificial muscle is excitation-contraction coupling. So there's a new channel rhodopsin that is highly selective for calcium, and Vlad has got this now integrated into the sarcoplasmic reticulum of these muscle cells. So the way we choose to mediate excitation-contraction coupling is we take the output of the electronic neurons and we use that to gate an organic LED. The organic LED will produce blue light, which will activate the channel rhodopsin to release calcium, and then we can use the normal troponin tropomyosin activation of the cross-bridge cycle to mediate excitation-contraction coupling. Now, on the sensor side, Dan has been working with epithelial nitric oxide synthase to get a nitric oxide signal, and he's got this fancy scheme by which he can shine light on eNOS that he's modified, and it will produce nitric oxide. And then Chris has been working on Luciferace Reporters, where he uses here a Rabanose to get luciferase to light up and produce different wavelengths. And the problem with this one is the kinetics are really quite slow for expression, and he's working around with something called the stressosome to try to find a way to speed this up. And we'll have more information about that later. Now, from my perspective, the really hard part was how do you get electronics that can operate on this scale? So I have a colleague, Young-Bin Kim, who does subthreshold analog VLSI, and we took the Marsh-Rose model and got it to operate in subthreshold analog VLSI with an 800 millivolt power supply. Now, originally we were going to use microbial fuel cells, and with 2 microbial fuel cells, we could get 800 millivolts. So we had adequate power to do that. And we can get these to produce bursting activity that we can modulate with inward current. And we also have a similar model for synapses, and we could get a inhibitory synapse pair to show alternating bursting, which is what we need to get swimming. And so the whole idea then is to integrate this into a 4-neuron central pattern generator, the motor neurons of which will activate the OLEDs to activate the muscle, and then chemical sensors or photosensors will activate command neurons to turn this whole thing on. And the idea was to get this to show chemotaxis, and I think eventually we'll be able to do that, but this is one of the beauties of this synthetic biology sandpit, and I will never forget this statement, is the sponsor said, if your project has greater than a 10% chance of success, we're not interested. So this is truly high-risk research. So I think we're still in the game. I think we can really pull this off. So Young-Bin Kim somehow got Samsung to agree to synthesize this circuit. And if you know anything about building subthreshold analog VLSI, a fab like this is about $150,000, and they've agreed to do 4 of them. So we even have a chance to debug it. So I think we're on the game for that. Now, how do you put this mess together at this scale? So originally we were going to make a robot that was 1 millimeter long, and we realized we couldn't get the electronics that small. So we're looking at a centimeter. Now, the standard way of doing this is with organic electronics where you use inkjet printing. Now, inkjet printing produces features that are on the order of about 100 microns. It's really hard to get a feature smaller than about 100 microns, and that's just too big for this. We fooled around with it. All the inkjet printers that you use are made out of plastic. If you try to use any fancy solvents, you melt the inkjet printer, which we did twice. It's really not fun. So we've gone to a new technology which is called electrohydrodynamic or EJet printing. And the way this works is you take a glass microelectrode, which is something that I really know how to work with, and you sputter coat it with gold, and then you put it over a stage that moves in X and Y, and then you put a very high voltage between the microelectrode and the stage of between 5 and 800 volts. And at the tip of the microelectrode, the ink forms what's called a Taylor cone. And then that Taylor cone calves off little micro droplets, which are about half the diameter of the tip of the microelectrode. So a good 30 megohm microelectrode has a tip diameter of about 1/2 micron. So that means that we can produce 250 nanometer droplets, which is pretty small. So that gets down into a scale where it really looks feasible to use this. And because we're using a microelectrode, which is glass, it's solvent agnostic. So we can use everything except for hydrofluoric acid, I guess. And so we can make inks that are nanoparticles of silver or gold or IPTO. We can make inks that have extracellular matrix proteins, viral particles with transcription factors, etc. So we really can begin to start to program the living part of this pretty well. So this is our EJet printer, and this stage here, which is the ultimate boy toy, it cost me $26,000, will move in 1 nanometer increments in X and Y. One wonders how you verify that, but it's pretty impressive. And it has on it a vacuum chunk where we clamp the substrate. We either use Kapton or cover slips for that matter. And then we have a light guide and a very high resolution microscope camera. And this is the business end right here. This is the stage, and this is a light guide and the high resolution camera. And so the idea is the shadow of the microelectrode, the distance between the electrode tip and the shadow tells you how far you're off the substrate. And these are little droplets of nanoparticles of gold and ethanol. And the ethanol evaporates almost immediately. And then you can make big splats to create pads for connecting wires. But we can produce extremely small patterns. And we've been fooling around with this now for about 3 weeks. What did I do? And already this is the kind of patterning that we can use. So these are traces that are 20 microns wide. These are 8 micron wide traces. But these actually conduct electricity, and these are made with nanoparticles here of silver. So I think the ability for us to be able to assemble and produce these things is really, really high. Now, how do you put this whole mess together? Well, first of all, we have this problem of excitation-contraction coupling. So the idea is we take a piece of Kapton and we print on it some anodes and ground conductors. Then we put on the conductive polymer layer of the OLED, then the emissive, the fluorescent polymer layer of the OLED, cathodes with independent conductors so we can control them independently, and then we coat it with a dielectric and with a muscle repellent, and then we put on polylysine and incubate it with myoblasts, and now we have a tail that has nerves, muscles, and an excitation-contraction coupling system. Now, in terms of the sensor part, we found a polymer photosensor that has the required sensitivity. It's a polymer mixture. We put then a layer of this polymer sensor between cathodes and an anode, and with a filter layer on this and a glue of polylysine, we can stick the cells on top of this and do the photo detection part of this. But to put the whole system together, we want to build all of this. We start off with a chassis. We put the OLED circuit on it. We put the microelectronic, the subthreshold analog VLSI, and we have a way of making a nitric oxide sensor using naphthalon and carbon fiber and platinum electrodes. And then we put a thin film battery on it and plate it with cells. And hopefully within 6 months or so, we'll be able to produce an actual biohybrid robot. Now, my long-term goal in this, being a neurophysiologist, is to get rid of all the electronics. And one of the big breakthroughs in the last few years has been the capability to form neurons out of stem cells and fibroblasts, and to do this in quantity. So what I'm really interested in doing is actually doing structural programming, where we form pads of trophic inks that will have viral vectors and transcription factors, extracellular matrix proteins, various glues, where we can put cells on them and transform them into different cell types. Then put readout circuitry where we could both stimulate and record and apply some of the learning rules that we know that mediate long-term potentiation and long-term depression. Then put guidance inks of semaphorins and materials like that that will cause cells to form connections between the pads, and then plate inducible neurons on them and actually form these CPGs de novo. So I think if we get to that point, then we can get the nerves talking directly to the muscles and really build a truly biohybrid robot using synthetic biology. So that's where I'm at.